Hello, this is Daniel Chiro. I'm back with another lesson in biology. This time we'll be able to look at the industrial application of enzymes. Before I go further with today's lesson, I want to remind you to subscribe to my YouTube channel. You can also leave the comment in the comment section below so that I get a response from you. You can also make suggestions on which lessons you want me to present. Okay, so let us discuss the industrial application of enzymes. One application is that enzymes are used or are included in the making of detergent powders. These are powders which are actually used for washing fabric, washing clothes. So you will be able to see that on the pack of a detergent powder you're going to purchase from supermarkets and different other shops. Those which contain active enzymes, it will be indicated on the pack that this detergent contains active enzymes. And the common enzymes which are included are what we call proteases, and these proteases actually will break down substrates which are, or let me say, they will break down the stains which are of protein nature. And the examples of stains which are of protein nature, those stains caused by blood, the blood stain on the fabric. You can have a chlorophyll stain on the fabric. These will actually be digested by the proteases, will be broken down into colorless amino acids, and so the stain will no longer be visible on your fabric. Then you will have washed it, because these are actually tough stains which ordinary detergents would not be able to remove. For example, if you are using detergent base, these will actually not be able to remove protein stains of the nature of blood or chlorophyll. So you need enzymes actively working. So for you to be able to do that, you need to ensure that the clothes are actually soaked in the detergent, and the temperature of the detergent solution you are going to make should be favorable, around 30 degrees thereabout, because the enzymes there will be able to act quite fast at such a temperature, between 30 degrees to 35 degrees Celsius. So on the pack it will be indicated the temperature at which the washing should be done. So you soak for at least 30 minutes to allow the enzymes to digest or to break down the protein into amino acids which are colorless. In that way, you will find that before you even do the actual washing with your hands, the proteins will actually be hydrolyzed to colorless substances, and you no longer be able to see the stains on the fabric. That is the common enzyme which is used, which is a protease. But sometimes you are going to find that certain biological detergent powders, because we call them biological because they contain enzymes. So these biological detergents may also contain enzymes such as carbohydrates, which will actually break down stains that are of a carbohydrate nature. Then they may also contain lipases, which will actually hydrolyze or simply break down the lipid, which can be a stain on your fabric. However, it is also important for you to note that carbohydrate stains, the lipid stains, are actually easy to remove even with ordinary detergent which does not contain enzymes. So that is why most of the detergents will only contain proteases, because protein stains are tough and they are actually difficult to remove until they are converted by enzymes to colorless amino acids. The next application of enzymes is in the brewing industry. So in brewing, enzymes are used, and we'll be able to discuss how these enzymes are used. So the first stage in brewing is actually you need to have cereal seeds. Cereal seeds contain starch in them. Okay, so the first stage is to initiate germination. So during that process, actually the seed has to be soaked in water, just enough water, not too much water, so that the enzymes that are present in the seed can be activated. And these enzymes which are present are actually amylases. And these amylases will act on the substrate present in the cereal seed, which is amylose. Amylose is starch. So the amylases will break down the starch to form maltose, which is a sugar. Now, this sugar is actually required for respiration, usually in the form of glucose. So another enzyme, which is called maltase, is going to catalyze or break down the maltose into glucose. And this glucose will be available for the germinating seed, so that the growth of the seed embryo inside the seed can begin to take place. And then you'll be able to see the radicle and the plumule pushing out. When they have pushed out, actually germination is stopped, because the sugars that have been made or that have been produced by the action of enzymes are needed in the brewing process. So the seed is the germinated seed, which is now called malt, because the main sugar which is present in there is maltose. Of course, some glucose will be present as well. So this is going to be dried. After being dried, it's actually ground. Grinding, I'm talking about grinding, it's taken for grinding, so that you form a powder, similar to the milling you use for making shima if you're in Zambia. Shima is simply powder. So when the powder has been made, it has to be made into a paste. So the process of making a paste involves that the water is heated. Before it begins to boil, the powder is actually added to form a paste. So that paste is boiled, and when it is ready, it is actually removed from the fire and cooled. After being cooled, the next step is to add enzymes, and these enzymes are added in form of yeast. So yeast actually contain enzyme Enzymes. So the enzyme Enzymes catalyzes or breaks down the glucose that is present in the paste and converts it to ethanol, which is an alcohol, the main component of alcoholic beverages. So it converts to alcohol, which is ethanol, and then a byproduct, carbon dioxide, is given out. So you'll be able to see bubbles forming, a process we call fermentation taking place. So in that way, alcohol is produced. So this alcohol can be removed if you want to make clear and not opaque alcohol. It's actually distilled off from the paste, more like in a similar way a brewing of the local cachasu, the common cachasu, which is actually brewed in the peri-urban and rural areas of Zambia. So that is one application of enzymes. So enzymes are applied in the seed when the seed is allowed to germinate to form what is called malt. Then again, enzymes are now artificially added by way of adding yeast, which contain an enzyme known as Enzymes. A similar application is also done in the baking. So in baking, actually what happens is that flour, sugar, and yeast can be added. We are talking about the baking which involves use of yeast, not where you are using baking powder. So yeast contain enzymes. So when the flour, sugar and yeast are mixed with water to form a paste, then kneaded very well to mix the components, what actually happens is that normally we form a substance which we call dough. Now this dough is normally covered so that you can allow the enzymes to begin to respire, and they respire without using oxygen. So when they respire without using oxygen, alcohol is produced, and we call that kind of respiration as anaerobic respiration. So they respire anaerobically, producing alcohol and carbon dioxide. So the important product there is carbon dioxide, which is going to cause the dough to rise, and once the dough rises, it becomes easier to cook inside when you are baking. So when it is being baked, the carbon dioxide actually helps to distribute heat inside the dough so that it becomes cooked inside. If you do not do that, it can be nicely appealing, looking brown, nicely, like it is nicely cooked. If there is no yeast to do that, or baking powder, you will not be able to enjoy eating, because inside it will still not be cooked, because the heat will not easily be conducted inside the dough. So that is the other application of enzymes in the industry. The next application of enzymes is in the making of sweeteners, sweeteners which are used for making confectionaries in the confectionery industry. You should be able to understand that confectionaries actually are sweet food products. So the confectionaries, normally the common sugar, the table sugar, the sucrose, is not the sugar which is used. So you need a sweeter sugar. The common sugar which is used is fructose. Fructose is actually made from glucose using an enzyme, and the enzyme that is actually used is called enzyme glucose isomerase. So the enzyme that is used is glucose isomerase. Why is it named glucose isomerase? It's because fructose is actually derived from glucose by simply, changing the position of certain atoms, and in that case, glucose or fructose is actually an isomer of glucose. By way of isomer, I mean that it is just the structure which looks slightly different, but the chemical formula is the same. So glucose has the formula C6H12O6. Fructose has the formula C6H12O6. But the structures, the arrangement of atoms in fructose and glucose is different, and that is why the two are said to be isomers of each other. So glucose isomerase is an enzyme which is actually used in converting glucose to fructose. The other application of enzymes in an industry is in the extraction of juices from fruits. Now, fruit juices, when extracting, if you're extracting juice from fruits at home, you don't get a lot of yield, you don't get enough from it. So in the industry, those companies that actually are in the business of making juices, which you buy, these juices actually are made to be produced in large quantities because they make use of enzymes, so that a lot of juice can be extracted from the fruits they are using. If it is oranges, pineapples, and so on and so forth, they use enzymes. And the common enzymes are cellulases and pectinases. Cellulase actually breaks down the cellulose, and it actually improves the yield of the juice. So there's more juice that is going to be extracted when the enzyme actually breaks down the cellulose. The pectinase is an enzyme that is used to digest what is called pectin. Pectin is a polysaccharide, and this polysaccharide actually causes the juice to form a substance that looks like jelly. So it's a gelling agent. So to prevent juices from gelling, pectin is added so that it breaks down the pectin. So that is another industrial application of enzymes. The next industrial application of enzymes is that enzymes are used to tenderize meat. So probably at some point you've gone to the supermarkets to buy the meat tenderizer. I'm not talking about the tenderizer where you hammer on the meat. I'm talking about the substance which is added to the meat to tenderize it, which is called a meat tenderizer in form of powder. So that substance, a meat tenderizer, contains an enzyme, and the enzyme is trypsin, which actually pre-digests the meat and makes it soft, so it becomes easier to cook. Ordinarily, you find that if you cook the meat, it hardens and it takes longer to become soft again. So if you want to cook meat fast, a tenderizer has to be added. In industry as well, during meat processing, tenderizers can be used to pre-digest the animal protein to make the meat a bit more soft. The enzymes are also used in making baby food. So you're going to find that normally babies have challenges in digesting. When they are beginning to be weaned, they are introduced to solid food, and some of the solid food may contain high protein content. So babies have problems digesting high protein foods. So to help them, the foods can be processed, can be pre-digested. So during the making of baby food, the food actually are present. You can purchase them in supermarkets. They are there. So this food, during the making process, actually proteases are added to pre-digest the meat, the protein which is present to form amino acids. So when the baby takes, they'll simply absorb into the bloodstream and begin to assimilate. By assimilating, we are talking about the baby, once the food is in the body, in the cells will begin to utilize either by forming other proteins or forming hormones and so many other products that can be formed from the amino acids. The other application of enzymes in industry is in what is called the tanning industry. Tanning industry is an industry where they convert animal skin, the hide, into leather. So normally you are going to find that because of the presence of the protein, once the animal skin dries, it hardens, so it is not flexible enough. So how that comes about because of protein, which comes to harden. So you cannot use such a skin. So when the skin has been removed from the animal, it must be processed in such a way that the protein is digested, and that way the skin, which now becomes leather, becomes pliable and very, very flexible. So you can make products such as shoes, leather belts, leather jackets, leather bags, especially handbags for ladies, and so on and so forth. So the enzyme trypsin is used to digest the animal protein, which is present in the hide of animals, the skins of animals. The other application of enzymes is that enzymes also used in the dairy industry. So enzyme rennin is used actually to process the milk, where the milk protein is coagulated, and this milk actually, the coagulated milk can be used for making products such as cheese. So we use an enzyme called rennin. Then lastly, the last application I want to talk about is actually in the production of sugar by sugar companies. Sugar companies normally extract sugar from starch by using amylases to convert starch into maltose, which is a sugar. So this sugar can also further be broken down to form, for example, if they want to make sweeteners as well, they can actually convert the maltose into glucose and then use glucose isomerase to convert glucose to fructose, which is a sweeter sugar, which can be used in the confectionery industry. Well, we have come to the end of this lesson. I'll see you next time.